Dear students, welcome to PUC second year biology class. We are studying the unit genetics and evolution. In that, one of the chapter that we were studying is principles of inheritance and variation. That the previous classes we have seen the inheritance of one gene or monohybrid cross experiment, which is being conducted by the Gregor John Mendel. He has concluded the result also, and suppose the result 1 is to 2 is to 1 was the result of genotypic which he has got, and 3 is to 1 was the phenotypic ratio that he has got, that we have seen. Mendel self-pollinated, and in the F2 generation, F3 generation, in the F2 generation, he found that dwarf plants continued to generate. And also dwarf plants in F3 and F4 generation. Not only that, but they start appearing in the F3, F4 generations also. That means concluded that the characters are hindered over there, suppressed over there, and they will be expressing in any of the generation. He concluded that genotype of dwarf was homozygous which has been appeared. And back crossed, he has checked it. Back cross, or it is also called a test cross. Back cross, crossing of F1 hybrid back with any of the parent. With any of the parent, crossing that one, and whatever the result that we will get it is back cross. And a test cross, crossing of F1 hybrid with its recessive parent. It is used to find out the unknown genotypic ratio. What was the unknown genotypic ratio? He wanted to know, and Mendel conducted a test cross to determine the F2 generation. He has taken the F1 hybrid with the recessive plant. He has done the experiment so that the result was 1 is to 1. He has got half of the flowers are violet and half of the flowers are white. So the result was unknown flower was heterozygous. The result is 1 is to 1 he has got. And this is one of the experiment test cross and back cross is being done for that. Mendel's principles or laws of inheritance. Mendel's principles or laws of inheritance. The first law is law of dominance, and the second law is law of segregation. The first one is law of dominance, and the second one is law of segregation. First law, law of dominance, says that characters are controlled by discrete units called factors. Characters are controlled by discrete units called factors. Factors always occurs in pairs. Factors occurs in pairs, and in a dissimilar pair of factors, one member of the pair will become the dominant. This is very important. And the other will become recessive. That is why it is called law of dominance. Dominant character is being expressed, and the recessive character is hindered, suppressed, not expressed. That is what it says. First law, characters are being controlled by discrete units called factors. Factors occur in pairs, always they occur in pairs. In a dissimilar pair of factors, one member of the pair dominates and the other is not recessive. That is recessive. This is the first law. And the second law is law of segregation. During gamete formation, the factors, or what we call them as alleles of a character pair present in parents, segregate themselves from each other such that a gamete receives only one of the two factors. Out of those two factors, one of the factor is being received. Homozygous parent produces similar gametes because similar type. If you see, for example, homozygous tall is represented with capital T, capital T. Capital T, capital T represent homozygous tall. Small t represents homozygous dwarf. If it is small t and capital T and small t, that represents heterozygous tall plant. So here, homozygous parents always produce similar type of gametes. But whereas heterozygous parents produces two kinds of gametes, each having one allele with equal proportion. They produce two kinds of gametes which are having one allele with equal proportions. This is law of segregation. He has given one of the law called law of dominance, in which characters are controlled by the discrete units called the factors, and those factors occur in pairs always. In a dissimilar pair of factors, one member of the pair dominates, what we say dominant pair, the other is called recessive. And second law is law of segregation. During gamete formation, the factors or the alleles of a character pair present in parents, they segregate from each other. They separate from each other such that a gamete receives only one of the two factors. Gamete receives only one of the two factors. Homozygous parents always produces similar gametes, which are similar. Heterozygous parents produces two kinds of gametes, each having one allele with equal proportions. This is the second law called law of segregation. And now you will be studying the dihybrid cross experiment, which is being conducted. Remember, this experiment is again conducted by the Gregor John Mandela. What he has called is dihybrid cross experiment. Dihybrid cross experiment. He has conducted this, and in this, two contrasting characters are being selected from each of the plant. From two plants, he has selected two contrasting characters. On that, he has conducted experiments for many times, and the result he has noted down and made the data. Mendel made some dihybrid crosses in pea plants. Example he has chosen is cross between pea plant with round shaped and yellow colored seeds and wrinkle shaped and green colored seeds. Taking into these two, he has crossed. You can see in the diagram, round shape is one of the character, yellow color is another character. Two characters he has selected from one plant, and wrinkle shape, the shape has wrinkled, and green color he has taken into account. So these two characters he has taken into account and conducted the experiment, what we call it as dihybrid cross experiment. Regarding the dihybrid cross experiment, further details we'll see in the next classes. So thank you, all of you.